Agriculture is an industry which employs more than 70% of India's population. Along with the large number of people involved in the industry, there is also a large number of diseases which affect the farmers. One such disease is aspergillosis. It is a disease caused by the fungus Aspergillus flavus and infects the farmer's lungs and thus causes symptoms such as coughing, blood, and also it acts like a carcinogen. So our research topic is to prevent and control aspergillosis using natural means. So our research question is to test the inhibitory effect of natural edible antifungal plant extracts on the fungal strain Aspergillus flavus. So why did we choose to research on this topic? Aspergillus flavus causes the disease aspergillosis, and as you can see in the picture at the bottom left, fungal spores are released into the air, and once humans breathe these spores, they sometimes grow on the lungs, and this results in fungal balls growing in the lungs. And at the X-ray pictures in the right, you can see fungal balls growing in the lungs. So another reason why we chose to do this is because antifungal plants are readily available, and the fungi can be cultured with ease. These are important as due to these factors we could do this experiment easily at a school level, and moreover it supports the idea of sustainable development, as instead of using artificial medicines we're trying to cure a disease by a natural means. So the three plants which we chose were the wild sage, the ginger, and the Indian basil, also known as the tulsi. So this is a list of the apparatus used, and we kept in mind that everything was kept as sterile as possible, since we could not risk having infections in the fungal culture. So this is the main apparatus used. The autoclave in the top left is used for sterilizing the equipment. Whereas laminar airflow provides a sterile environment for the transfer of fungal cultures. So now I'd like to move on to the methodology and hand over to Ishita.
Okay, so the first thing that we come to in the methodology is the making of agar-agar solution. Agar is a special nutritive medium for the growth of fungus. So the one that we chose here was the potato dextrose agar, or the PDA as we call it. So the making of the PDA was as follows. So first we took 100 ml of distilled water and we heated it till it was lukewarm. Then we added 10 grams of PDA and in the lukewarm water so that it mixes well. Then we added another 150 ml of water in it, and then we autoclaved it at 121 degrees Celsius for 15 minutes. So it ensures that the PDA solution, which is this one, it is heated properly and it does not settle in at a gelatin, like a gelatin. So after that we put it in the sterile petri dishes under sterile conditions. So we left it at room temperature for 10 minutes, and after 10 minutes we saw that the gelatin has, the one that here, so we saw that the gelatin, the PDA, had settled down as a gel structure, and so we kept it in the sterile conditions until the fungus had grown and was ready for inoculation. So we come to the growth of fungus. So we took a tomato, we sterilized it with ethanol, and then we cut it and left it exposed to air. After 5 days, we saw fungal growth on it. So we actually inoculated that fungal growth into the test tubes. So after 5 days, the fungus had grown on it, and after this, we inoculated the fungus from this test tube into another petri dish for inoculation purposes, into other different petri dishes. Preparation of plant extracts. So the plant extracts that we have is the one with the ginger, the tulsi, and the wild sage. So for ginger, we actually took the rhizome and we peeled it and we crushed it, and then we boiled it. So to get the extract. So we took 1000 liter. So we took 1000 ml and we boiled till it came to 500 ml. So we get the concentration, and in case of wild sage and tulsi, we took the leaves of the plants, we cut them, we washed them, and then we put it in 1 liter of distilled water, and then we boiled it till it came down to 500 ml. And then we refrigerated them till the fungus had grown. Now, inoculation of Aspergillus flavus. After the fungus had grown, we inoculated into 12 petri dishes. So, there's the disc borer there. We chose this device so that we could get a particular diameter for the inoculation. So, there's a pure culture over there, and then we took the fungus from the pure culture and we added it into a new sterile petri dishes with agar into them. So you can see us inoculating, and this is the inoculated one. And we chose 3 petri dishes. We chose 12 petri dishes and we inoculated into 12 petri dishes. So yeah, this is the one that we did. Now, addition of plant extracts. This is the ginger, this is the tulsi, and this is the Lantana camara or the wild sage. So we actually chose we took the help of micropipette to transfer precise amount of liquid that we needed. We took 1000 microliter of plant extract, and we actually set it to 500 microliter, and we took it twice from this, and we added into the petri dishes as the freshly inoculated petri dishes here. Yeah, so now my friend will talk to you about the results.
Thank you, Ishita. So as you can see the results here, after we left them for a period of 5 days, the results over here, we can see them as a control. Thank you. This is all the growth of Aspergillus flavus, which has covered the entire petri dish. Zingiber officinale, which is the ginger, is almost as much as the control, but a little lesser. And hence, we see that it's not very effective in preventing fungal growth. In Tulsi, or the Oxymum sanctum, there is a little bit of growth, but comparatively lesser, so it is quite effective. But Lantana camara, we saw, was the most effective, as you see no fungal growth at all. And this is another reading that we took, and we saw similar results as well. At a concentration of 40 grams per liter, this is what we achieved. And these are closer pictures of control with ginger, as you can see, control with tulsi, and control with Lantana camara, or the wild sage. As you can see, there is no growth over here. So our data analysis was that the original diameter taken by the disc borer was 0.5 centimeters. And after the growth of 5 days, we observed the growth of the control to increase to 0.78, for ginger 0.75, for tulsi 0.60, and for lantana 0.42 centimeters. And therefore, the percentage change, as in the bar graph, we see is this: 56% increase for the control, then 50% increase for the ginger, 20% for the tulsi, and minus 16% for the lantana. Lantana, we found, was so effective that it actually prevented the fungus from growing because it killed the spores itself, and it reduced the inoculated fungus diameter to 0.42 centimeters. So, on seeing these results, we decided to research and search upon what actually caused the fungal growth to stop, especially for wild sage or Lantana camara. It contains a few chemicals from the essential oil, which are mono and sesquiterpenes. And fungus grows by producing a gastrolol. It is a chemical compound which controls the fluidity and the plasma membrane of the fungus. And we saw, and as we read, that this chemical, it creates lesions in the plasma membrane and hence reduces the amount of gastrolol produced and hence kills the fungus. We also had a few limitations in our project, and you can see there is a small scale contamination here with 3 other Aspergillus species in a petri dish. And also, as aspergillosis is a disease which affects the lungs, we were not able to simulate the conditions of the bronchioles. And also, to maintain the temperature conditions was quite difficult. Therefore, we conclude that all these 3 plant extracts, ginger, tulsi, and the wild sage, have antifungal properties, where Lantana camara is the most effective, while ginger is the least effective. And also that it is a natural way of curing a disease. But at a school level, we cannot prove this in a clinical way, but it can be an alternative way in the future for treating such a disease. So therefore, this was our project, which we had positive results for. Next question, please.
Hi, I'm Sandra Long from St. Paul's Educational College. Since the fungus actually infect the lungs instead of the stomach, so do you recommend the patient to breathe in the essential oil of lantana, or do you prevent it?
Yes, in this way, we actually suggested 3 methods. One can be an oral consumption of this, because if it gets digested, the chemicals will come into your blood. And as there is also blood flow and capillary networks in the lungs, so it will automatically be present there and it will infect the fungus. And also, yeah, we also suggested that we can inhale on boiling, we can inhale the fumes, or we can even take nebulization for the lung problem.
This is just a clarifying question. You measured, I thought you measured the diameter of and the diameter change. Can you just go back to the slide and show us where that diameter was? So on the photo of the plate, so you're measuring the diameter of that centre. Can you show us the diameter measurement there on the photo? So it's that little spot in the middle that you mentioned the diameter, thank you.
That was the inoculated fungus, and what you see has been the sporing growth, as in it has released its spores, and now it's growing all over the petri dish.
Okay, but the actual measurement you had in the table was still just the diameter of the...
As far as my knowledge is about lantana, the government of India is not promoting lantana, because where lantana grows, no other vegetation takes place. Have you thought about this? We have lantana all around Uttarakhand, and it's dangerous for all our vegetation.
So yeah, lantana is dangerous for several local species, and there are negative effects of lantana.
All species, or no vegetation grows, even paddy fields they are destroying.
So at least of the lantana plants which are remaining, we can utilize the positive aspect of the plants by and we can use the positive effects which actually help us in a medicinal way, instead of worrying about the invasive effects as of now. That's what we are thinking.
And also we researched, and we had to look at a few research papers, and that said that the leaves aren't poisonous, but its berries and seeds, they are poisonous.
Berries are not poisonous, berries are bird attracting.
I'm sorry, but I think I read a report once that which had caused the death of a few children, so I would go by that. But I have been told that yes, sometimes they aren't poisonous, so we were still researching on that.
Okay, thank you.
I was just wondering, what's the survival rate of this disease that you're running? Like, how many people die if they get it?
Not all people die when they get it, but the situation is quite bad because you actually have fungus balls growing in your lung, so you tend to spit out blood, and Aspergillus flavus also releases a chemical within your lung called aflatoxin, and this is a carcinogen. Yeah, so people don't die immediately from it. However, in a long-term process, yeah, they tend to die out quicker.
And also, as it locally mostly affects farmers because it affects agriculture, especially the peanuts. So, and farmers do not have that high an income. So getting the treatment done, getting the hospital aid and the payment is also difficult for them. That's why.
That's it. Thank you.